In this video, we are going to talk about genome composition of plant, animal, insect and microbial species. The structure of organism's genome varies according to the domain of life it belongs to. Prokaryotes, which include both domains of bacterium and archaea, have small and highly energy efficient genomes, typically consisting of few million base pairs. Eukaryotes, on the other hand, have genomes ranging from the very small, approximately that of the larger bacteria, in example Encephalitozoon cuniculi with its almost 3 megabases genome, to the very large, consisting of over 100 billion nucleotides, such as the lungfish, Protopterus aethiopus, and the monocot plant Paris japonica. Base composition is generally more similar within closely related groups and organisms residing in the same environment. For instance, the average genomic GC content of the currently sequenced avian, mammalian, and reptilian genomes all lies somewhere within 40 to 50% GC, with GC3 slightly higher. GC content in bacteria and archaea range from approximately 13 to 75 GC, and genomic GC percentage correlates strongly with GC3. Whereas the genome size of mammals is somewhat larger than that of reptiles and birds, roughly 3 gigabases versus 2 gigabases, they have far from the largest genomes. Several plants and fishes have substantially larger genomes with more protein coding genes than mammalian genomes. The genome size doesn't increase with the complexity of organisms is known as the C-value paradox. Some model organisms' genetics are well studied and they will exhibit characteristics that represent a larger group of organisms. We will now show more characteristics of their genomes, of their kingdoms, and the domains.
Okay, I'm going to talking about plant genome composition. Until very recently, the molecular analysis of plants often focused on the single gene level. Recent technological advances have changed this paradigm, enabling the analysis of organisms in terms of genome organization, expression, and also interaction. The study of the way genes and the genetic information are organized within the genome, the methods of collecting, analyzing this information, and how this organization determines the biological functions is referred to as genomics. And also genomic approaches are permitting every aspect of plant biology, and since they rely on DNA coded information, they expand molecular analysis from a single to a multi-species level. Plant genomics is revising the previous paradigm of identifying genes behind biological functions, and instead focuses on finding biological functions behind genes. And also it reduces gap between phenotype and the genotype, and it helps to comprehend not only the isolated effect of a gene, but also the way its genetic context and the genetic networks it interacts with can modulate its activity. And in next slide, we can see the genetic structure of plant genomes. For example, as you can see in this table, plant genomes are best described in terms of genome size, gene context, and the extent of repetitive sequences and the polyploidy duplication events. Also, plant also possess mitochondrial and choroplast genomes. The nuclear genome is the largest and the most complex. There is extensive variation in nuclear genome size, as you can see in the table, without obvious function significance of such variations. Plant genomes contain various repetitive sequences and also retrovirus like retrotransposons containing long terminal repeats and the other retro elements, such as long interspersed nuclear elements and the short interspersed nuclear elements. And also these retro elements is insertion contribute to the large difference in size between collinear genome segments in different plant species, as you can see in this picture, and also 50% or more different in total genome size among species with large genomes, such as, for example, corn. And the next is animal genome composition.
Okay. We know that an important key to more specific and effective animal breeding in the containing within our knowledge of genomes. And nowadays, researchers have the sequenced and the complete genome of the 100 of the animals and the plant more than 250 animal species and the 100 species of the bird along the list, continuing to by the grow almost a day. Firstly, we know at an example animal composition, you have the rat genome, and the rat genome, the recombinant finding a genetic variation measure of the DNA level. It's 2 or 3 large within the most completing. And the rat genome have the 2.57 gigabyte and with the smaller than the human genome. Human genome have the 2.9 million base pair, and the global opposition of the 3 genome reveals the large chromosomal revisions. Refer to the orthogon with the chromosomal segments, which have been the inherit with the minimal rearrangement of the gene order from the primate, rodent, and hedger. How we see in the picture, the first rat genome they applied the research in the 1828, and the human rodent they derived from the common enhancer 80 million years ago. Rat and the mice derived 12 and 24 million years ago, and the mice genome have the 2.6 billion base pairs. And what about the second animal's example? It is a pig. And the illustration of the genetic information of the breeding of the rat and the genomic selection and use it for the fixed selection against broad training. And we have the most of the opportunities of the genomic selection of the difficult terrain and the available of the field health and the methane emission for the cattle sensitive and the climate and the product quality. The genome of the pig, the chromosome of the 18 with the X and the Y, and the genome size the smaller than the human, and the 2.7. The pig, a member of the archaeopteryx, and which means the cloven-footed dead mammal, which and the evolutionally clade the distinct from the primary of the root. And mainly the chromosome would have the parent from the next generation selected based on the single nuclear polymorphism genotype, which have the translated the predictive breeding words based on the information from the trading population. By this way, will be the select the pig's genome. The next one about the insect.
In nuclear genomes, in insects consist of the chromosomes consisting of DNA and the proteins. And the nuclear genome is the largest contributor of genetic information within an insect. And here the mitochondria, which are organelles in the cytoplasm, also are components of the genome. And the mitochondria are derived from the bacteria that they became essential symbionts of eukaryotic organisms, and they contain a number of genes essential to the insects. And finally, in addition to the nucleus and the mitochondria, many insects contain intracellular and extracellular microorganisms that provide essential services to the insect. Symbionts may be bacteria, viruses, fungi, or spiroplasms that live in or on their insect hosts. And many insect symbionts are unable to survive outside their host, and many insects cannot survive without the services of their symbionts. Thus, the insects contain genetic information from the several sources. And also, there are two main factors that affect the quality of insect genome assembly: the repetitive sequences and the heterozygosity. And a large number of repetitive sequences in the genome can cause the substantial ambiguity in the process of assembling contigs and scaffolds. And the heterozygosity or allelic variation in sequenced individuals also greatly complicates the problem of genome assembly. And currently, the 100 to 119 insect genome sequencing projects have been registered with the National Center for Biotechnology. And they are including the 401 that have genome assemblies, and the 155 they're annotated that the protein coding genes.
So, talking about the microbial genomes, they are widely variable and reflect the enormous diversity of bacteria, archaea, and lower eukaryotes. Bacterial genomes usually consist of a single circular chromosome, but species with more than one chromosome, linear chromosome, and combination of linear and circular chromosomes also exist. The smaller microbial genome is the Mycoplasma genitalium, which encodes 525 genes, and some of the largest ones is cyanobacteria, which encodes more than 12,000 genes. So one major difference between the genomes of microorganisms and higher eukaryotes is the presence of circular extrachromosomal DNA, called plasmids. Plasmids can be transferred via horizontal DNA transfer from one cell of the same generation to another, mediating the rapid evolution of many different organisms. So the study of microbial genomes help us to better understand the broader biology of bacteria and how their genetic composition contributes to their tangible characteristics. The study of genomics is also important to infer the evolution of bacteria. Bacteria often evolve not just through small single nucleotide level changes, but through the quantum evolutionary events. These include through the transfer of plasmids between species and also the transposition of large genetic elements within single cells. Understanding this process allows us to determine the origins of bacteria and map the transfer of genes such as those conferring antibiotic resistance.
In conclusion, the average genome sizes of the 6 kingdoms and subkingdoms that were showed are in order from the smallest to the largest, like archaea, then bacteria, then protists, fungi and plants, and then animals finally, with the largest genomes in average. Microorganisms and higher organisms showed very different patterns of C plus G content when the genomes were ranked by size within each large phylogenetic group, and the genome level average C contents were ranked according to the percentage of the total CG nucleotides in the total genomic nucleotides of the kingdom or subkingdom, with the result being as follows. Protists have 26 and 42%, plants have 41.10%, animals 41.20%, archaea 44.88%, fungi 47.96%, and bacteria with 50.76%. And as we march down the path of deciphering more and more organisms' whole genome sequences, genomic analysis has and will continue to shed new light on the evolutionary and economically important genomic components across the kingdoms, as well as refinement of the current phylogenetic tree obtained by fossils and other methods.
Thank you for your attention.
Thank you for your attention.